Dear students, in this module, I'm going to talk about the protein structure comparisons. You know that the proteins are progressively assembled into primary, secondary, tertiary, and quaternary structures. The primary structures being the amino acid sequence in the protein, while the secondary structure being the chain of amino acids. When it gets folded onto itself, then it takes the shapes of alpha helices, beta sheets, loops, and coils, while the tertiary structure is essentially a combination of the secondary structures, and the quaternary structures, of course, are the combination of different proteins interacting together. So if a protein has this organization, then what does it give rise to? It gives rise to specific function, and that the structure is a determinant of the protein function. As I just mentioned, that the function can be calculated by simply looking at the structure of the protein. Therefore, to compare two proteins would mean that if you know the function of a protein and you have a protein with an unknown function, and if they have the same structure, then you can also predict the function of the unknown protein. So in this way, if you are given a capacity to compare two protein structures, then you can calculate the function of the unknown protein. More so, there are two types of comparisons. The first one is that if you compare the entire protein structure. So this is called the rigid body superpositioning. So the proteins are considered as rigid, and you have to superimpose them onto each other and see the differences. Secondly, there is the partial comparison. So first we will look at the whole protein structure comparisons, and later we will see the partial structure comparisons. So for comparing the whole protein structures, you have to compare the secondary structures, the motifs, the domains, and the organization of each of these within the overall protein structure. So once the entire protein structure is comprised and constituted, then you can compare it with the other unknown protein. So the structures of all such subunits are to be considered as one and then compared. Now towards comparing portions of the entire protein. So there may be cases when the two proteins are not matching to each other, but that portions of these proteins are very similar. One example can be the domains. As you know, that a protein may contain multiple domains, and the domains are functionally independent subunits of a protein. So such domains may be shared between different proteins which are performing the same function. So therefore, there is a good case to compare subunits or partial structures from the overall protein structure. For that, you can simply compare the motifs and domains. So, if you are given a task to compare the partial sequences, then you simply extract those portions from the protein structure that are of your interest, and then you compare them using rigid body superpositioning. We'll talk about this in detail in the later modules. Now, given that you have two proteins to compare, or let's say two domains to compare within the bigger proteins, then how to compare? How to do rigid body superpositioning? The clue or the answer to this question is that we have the alpha carbon coordinates of every protein in the PDB. So if you have a protein with its PDB in the structure databases, then you can extract the alpha carbon coordinates, that is the X, Y, and Z position, and then compare them with the alpha carbon positions of the other proteins. So let me help you to remember what the alpha carbons were by looking at this figure. you see that there is this backbone of the protein and it continues onwards, and here are your side groups, and beneath these are the alpha carbons here. So if you have the coordinates of these alpha carbons, then you can compare them with the positions of other alpha carbons in the other protein. Now, if you want to compare the two proteins using their alpha carbon positions, then let's consider this example. So here I have plotted the alpha carbon positions in a protein's backbone like that. And as you can see that each position can be compared with its relative position in the other protein. Similarly, you can compare the rest of the alpha carbons as well, and eventually you can talk about their similarity. In this case, it appears that the two proteins are exactly similar. In fact, for you, this is the same protein in both the figures. So in conclusion, the PDB coordinates of the alpha carbons are used to compare the distance between the two proteins. And then we have the whole protein structure that is compared, or even the domains.